This is the second part talking about Mendel's experiments. In this part, we're going to actually continue on with dihybrid crosses and special cases of inheritance. So let's go ahead and first start talking about special cases of inheritance and the goals and objectives we're going to actually achieve at the end of this lecture. We're going to be able to identify non-Mendelian inheritance patterns such as incomplete dominance, codominance, and sex-linked inheritance, as well as we'll be able to explain what pleiotropy is and epistasis. So let's do a little recap of what we learned in the last lecture. We learned that when we're talking about genetic crosses, which you all did an example of a monohybrid cross, that the P stands for parents of the, and those are parents of the F or filial offspring. We also learned that F1 is the first generation offspring, so F2 would be the second generation offspring. And that is, if you cross two F1 offspring together, that is what's giving you that F2 generation offspring. So now we're going to go ahead, we already learned about monohybrid crosses, we're going to learn about dihybrid crosses. Remember, monohybrid means that we're crossing for one trait. Dihybrid, di means two, it means that we're now crossing two different traits in one cross. So for example, we actually have here, this would actually be indicating our yellow seed color and our green seed color. Also, we're crossing the round trait versus the wrinkled. So we're crossing two different traits at one time. That's why we have yellow and round versus green and wrinkled traits. So what happens if we cross these guys individually together first? So to get the actual gametes for each of these, we would have to do kind of like the FOIL method, where you have big Y, big R, that's one gamete. Then again, you would do this big Y, big R, that's the second gamete, and the same thing over here, big Y, big R, big Y, big R. So as you can tell, these are all the same gametes. That's why it was very easy to understand and very easy to just do this cross really quickly, because it's the same thing over here. We have little y's and little r's. So they're only going to have one combination of gametes that they can deliver. When you cross both of these traits together, you're actually going to come out with this heterozygous individual, and this individual is heterozygous for both traits here. So they're heterozygous for the actual yellow seed color, because you see big Y, little y, and they're heterozygous for the round seed color, because it's big R, little r, and this would be considered our F1 offspring. If we cross two of these F1 offspring, however, we're going to have a whole new outcome. So do the FOIL method again. So we're going to have big Y, big R. That's going to be our first gamete. We would also have little y, little y, big R. That's our second gamete. Then right here, you're going to actually have a big Y and little r, because we haven't done that one yet, and also our little y, little r, our fourth gamete. Since these are both heterozygotes and they're the exact same individual, we know that these gametes will be the same that these gametes have on this side as well. So monohybrid cross, remember we had 4 squares. In a dihybrid cross, you're going to have 16 squares to look at those F2 offspring with. And you guys can follow along. You can go through and you can see the individuals here. But all in all, they actually color-coded this quite nicely for you, so you can actually see the phenotypes of the offspring. So yellow and round are going to be any individual that has a big Y or a big R in it. So if you follow along, go through all the 16 squares, you'll see the ones that actually are yellow and round phenotypically. There's a total of 9. Then we're going to have 3 of these other kind of offspring, and then 3 of these right here, and then only 1 of a wrinkled and green offspring right here. So it's actually known as a 9:3, 3:1 ratio phenotypically. It's going to have a different genotypic ratio, but to make things simpler, in this case, we're going to just look at the phenotype. So individuals that breed true for 2 different traits are crossed, and that's exactly what it's showing right here. That's all this is saying right there. This is just that part highlighted and enlarged for you to actually see. So this is just exactly what Gregor Mendel did. He was just trying to figure out what was going on and see if one trait would actually mask another. So would the yellow or would the seed color mask the wrinkled or the round seed, the actual shape of the seed? So that's what he was looking for here, and that's where he discovered the 9:3, 3:1 ratio right here. As you recall, one of the principles that Mendel came up with was Mendel's law of independent assortment. The law of independent assortment states that allele pairs separate independently during the formation of gametes. This means that traits are transmitted to offspring independently of one another. So this can actually occur when alleles are on different chromosomes, as you can see right here, because crossing over, they're not going to affect one another at all. And also when they are on the same chromosome, but when they are far enough apart to still allow for crossing over to occur. So we're going to pretend right here is a chromosome that's crossing over with this one right here. So as you can see, the X is going to cross over right here, and the gene 2 will be interchanged with this other homologous chromosome, but it's not going to affect gene 1. So they're going to still go ahead and assort independently of one another of different genes. Linked genes, however, are known to happen or known to occur when the actual genes are very, very close together on the same chromosome. For example, here. So if I had crossing over same thing right here, as you can see, the variation or the actual exchange of information would actually occur, and maybe both gene 1 and gene 2 would actually be grabbed and switched out with the other chromosome. So that's kind of affecting the way that they should be assorting independently of one another, and that's just due to their loci being so close together. That's why they're known as linked genes right there. So let's go ahead and look at an example, and let's try to do a dihybrid cross. In humans, brown eyes is dominant to blue eyes, and brown hair is dominant to blonde hair. So you can already go ahead and start and make your key there. A woman with blonde hair and blue eyes, so what I would do first is go ahead and draw out your genotype for that woman. And then a man who is heterozygous for both traits, go ahead and draw out your genotype for your man. How many of their children can be expected to have blonde hair and brown eyes? So that is going to be a dihybrid cross, so you should end up with 16 squares at the very end of it. So here's going to be the alleles I would have chosen. Just like I said, you could use any letters you'd like, any ones you feel comfortable with, and it should come out with the same outcome, so don't worry. So remember, the question is asking us how many of their children can be expected to have blonde hair, brown eyes. I'm giving you the alleles. Go ahead and work on your own and start. And if you'd like to check back, you can email me, or you can check with a tutor and find out if you're doing the work correctly on this part. So let's go ahead and also talk about what the product rule is. The product rule is when you actually want to find out the probability of two or more independent events occurring together, and that can be calculated by multiplying the individual probabilities of the events. So in this example right here, we want to know what's the probability of getting a heterozygote for this B and also a heterozygote for this CC. Because remember, this is one trait being crossed and this is one trait. So we want to find out what's the possibility between these individuals of us getting that. So what we're going to have to do is we'll cross this individual by itself with that trait itself. So we're going to cross this big B, big little b, and this big B, little b. And we're going to go ahead and do fur color over here. And let's say the little c's and the big c's are going to be with fur texture. So you just go ahead and do your crosses of those individuals and their alleles, and you come out with a 1/2, because it's 2/4 for this big B, little b. And then you have the chances of getting big C, little c also 2/4 or 1/2 of the time. So all we have to do is now multiply this actual probability of this occurrence and that probability of that occurrence, and we get 1/4 is the probability, or 25% chance probability, that we can actually come out with those two independent events occurring at the exact same time. That's a little difficult to understand. Think about it with dice. So for example, let's say we want to get a 6 and we have one dice. one die. So I'm going to go ahead and roll a die. What's the chances of me coming out with a 6? Well, there's only one face of the dice that has a 6 on it, so I have a 1/6 chance of getting a 6. Let's say I roll 2 dice at the exact same time. How would I find out the probability of getting a 6 on the one dice and a 6 on the other dice? So you would actually multiply 1/6 times 1/6, and you get 1 out of 36 probability chance of those two independent events occurring at the same time. Independent events occurring at the same time, whenever you see word problems with where you would have to multiply probability, you're going to see the word and. So what's the probability of me getting a 6 on a die and a 6 on another die? That's what they mean by that. Okay, and those are the examples we just looked at. So now we're going to talk about different types of inheritance. Now these are non-Mendelian inheritance that we're talking about. Before we did talk about complete dominance, that's Mendelian inheritance. I just wanted to recap on that. Remember, that's when an allele is basically is regarded as dominant and it completely masks a recessive allele in another case. So let's say big A will always completely mask little a. That's complete dominance. And we have incomplete dominance. That's where a phenotype of a heterozygous organism can actually be a blend. So we'll talk about all of these individually in just a second though, so don't worry. And codominance is where both alleles are expressed in the phenotype, and they can also be considered sex-linked, which is a gene is actually located on the X or the Y chromosome, but mainly we're going to be looking at it on the X because the X has so many more genes than the Y. So we usually are going to be looking at problems with X-linked inheritance. So here we're going to look at complete dominance first. What is complete dominance, okay? It's when a single dominant allele causes a dominant phenotype to be viewable. So it's all the kind of examples we've been looking at. So whenever you see a capital letter, it's going to completely mask whatever phenotype the little letter or the recessive allele is going to be showing. So in this example, we have right here a homozygous dominant flower, which is the purple one. That's how it's big T, capital letters. And that one is crossed with a white homozygous recessive flower, which is the little t. When you cross these two together, the purple flowers or these dominant alleles completely mask the white alleles. So even though these are all heterozygous, it's big T, little t, big T, little t, big T, little t, and so forth, no matter what, it's still going to be the purple big T is going to be masking the little t. That's completely dominating another allele. The second kind of inheritance we will be looking at is incomplete dominance. I like to refer to this as a blend or a blending of different alleles or different traits. So it's like the example here I have is actually an ice cream cone and a light bulb. And it's kind of like if they made a blend of an offspring, then you would get that spiral light bulb that we see at the stores. Or another way to think about it is if a fork and a spoon come together and copulate, then they're going to be actually making a spork. It's a blend, so they're not going to be more like the mom or more like the dad. They're going to actually make a complete new intermediate phenotype. So one example is you want to refer to is snapdragons. Snapdragons, basically the dominant allele, or let's say we're still talking about the big T, right? The big T, when it's actually crossed with the little t, when it becomes a heterozygote and it makes that big T, little t, it's actually going to be a brand new color of flower. So if we were doing the purple and the white, then it would be like a very light lavender. It's going to be a blend of those two traits, okay? In snapdragons, like we see right here, if you cross the red with this white yellowish snapdragon, it makes a pink, an intermediate phenotype. So once again, incomplete dominance is when we are crossing two true breeding plants. So that's be all red plants or all red flowers crossed with the all white flowers. That is what we said was going to actually give us our third intermediate or blended phenotype, which would be our pink in this example. So let's go ahead and actually go through an actual cross of one. So right here, all I'm doing is a big C to keep track of. I'm talking about the color trait, the color of the flower in this case. So if I'm doing the little r's as my alleles, so these are my recessive alleles, this would be my white. If I am doing my red, then I'm going to have my capital alleles, which is R. But remember, this is not a dominating allele here. So the big letter is not going to dominate the little letter in this case. They're going to blend together and make a new intermediate phenotype. So when we do, I set up our Punnett square. Right there would be our parents. So our red snapdragon crossed with our white snapdragon. And now we're going to go ahead and do the cross, follow it through. So big R and the little r come down. Same thing with all of the rest of them. As you guys can see, we're going to make 4 offspring. And all 4 offsprings are heterozygous because it has big R, little r. And that's going to result in our intermediate or our new phenotype, which is a blend of the parents. And that's why they all end up as pink flowers in this case. So now let's see what happens if we cross 2 of our F1 offspring from our previous question we were looking at. First thing we're going to do is write out our legend or our key so we can keep track of what all the different alleles and combinations stand for. Right here we have the white, the red, and the pink snapdragons because we know it's incomplete dominant, so we know it can actually make a blend of those 2 alleles together. So here's going to be our first parent and those 2 gametes. Here's our second parent with those 2 gametes. And when we go ahead and do the cross, our resulting offspring is going to be, right here we have a red snapdragon, 2 pink snapdragons, and a 1 white snapdragon. So this is going to actually be a 1 to 2 to 1 ratio in reference to their phenotypes. Genotypes, it would be the same thing in this case as well, but just you're going to actually write out the genetic makeup of each individual. So once again, 1 to 2 to 1, and then phenotypically 1 white, 2 pink, and 1 red flower. So let's look at an example of a certain question you might get on an exam, something you might want to be able to work through. Okay, a father has curly hair and a mother has straight hair. How many of their children might have wavy hair? So obviously there's actually an intermediate phenotype here, which is going to be our wavy hair. So straight and curly make wavy. So we know it's an incomplete dominant situation. So first things first, we're going to go ahead and write out our key. I just happen to use H's, the big H, because I wanted to stand for hair color, but you can choose any letter you'd like. It's going to be the same result. Then I did the actual alleles C for curly, I did S for straight, and then I did, well, of course, when these 2 mix together, they're going to make that new phenotype, which is going to be our wavy. And you all can go and actually work that out on your own and see if you can get it. If you need any help, email me, message me. I'm glad to help you. And even if I have to make another video for you to see or understand, I'll be perfectly happy to do so. So here's another type of inheritance. This is called codominance. Now, incomplete dominance, remember, it's a blend of the parents. Codominance, however, though, is actually going to be different. This is going to be where 2 alleles are expressed equally in the phenotype, but they're not going to blend together. So previously, in incomplete dominance, we had red flower and white flower made pink. In codominant, they're going to codominate together, which means they both show up. That's why right here in this example, they are actually going to both show up. So we have red and white petals, and they both show up equally in the actual new offspring. It's not a blend, so it's not a pink flower. It's actually red and white petal flower now. That's codominance. So another good example of knowing what codominance means, that means that, or a good example to know would be human blood types. Because you have the A blood type, you have the O blood type, you have B blood type, and you have AB blood type individuals. So these are the different ways you would actually write out the genotypes of each of these. And these mean the exact same thing, so you can write them out either way. You'll come out with the same exact answer. I just wanted you guys to actually visually see how they actually write them out, so you can actually be familiar with them. So blood type O right here actually does not have any antigens on its red blood cell, so that's why there's a little I right here. That just means that it doesn't have any antigens, therefore it won't make any antibodies towards anybody. So that's why they're the universal donors. Okay, down here, if you look at this is why I have this one highlighted. If somebody is type AB, that means that they actually have the A allele and the B allele. and they're both codominating together. That's why their blood type is AB. So they're not masking one another. They're not a blend of each other. They're actually both going to show up because on the red blood cell, they actually have antigen A and antigen B. So this is what I'm talking about when I'm talking about antigens. If you are blood type A, for example, let's look at this part right here. Let's look at this whole column. If you're blood type A, what that means is that you have antigen A's on your actual red blood cell. What these guys do is they actually tell your body when there's an actual invader in your bloodstream, because they don't want anything that shouldn't be there to be there. So they're kind of like hey, we have an invader. They're like the guys that are going to trigger your immune system to come in and take care of any business if any kind of bacteria or any other antibody showed up. So what they're responsible for doing is making anti-B antibodies. That means that it does not want anything to do with type B blood. So if it did, let's say, for example, let's say a nurse made a mistake. Let's say you were supposed to get a blood transfusion. You should only get blood type A or blood type O, because what's going to happen if they give you blood type B into your bloodstream, well, you already have these antibodies, right? So your antibodies that are naturally made in your bloodstream are going to sit here. Look how this one's formed like a triangle, and look at antigen B with people with B blood type. These guys are going to actually attach and attack that B blood that gets transferred to you by accident, and unfortunately that's going to actually coagulate your blood or cause it to get all clumpy and meshed up. But that also is a fatal condition, because you can't have your blood not circulating like it should, and as quickly as it should, and as efficiently as it should. So as you can imagine, it's going to lead to a lot of major issues. So people with blood type O, they are the universal donors, and people with blood type AB, look at this, they have both antigens. That means they make no antibodies. So that means they are the universal receivers. They can receive blood type from A, they can receive blood type from B, they can receive blood type from O. There is nothing that they're going to be fighting against, because they can receive all of the other blood types. So here's an example for you all to look at and try to work through, okay? A woman with type A blood has a child with type O. The man she claims is a father has type B blood and is homozygous. You are the judge, so please tell us, is this a child's father or isn't it? So I did give you guys the phenotypes and genotypes right here, so you can go ahead and you don't have to write these out. This is going to be the key you would start with though. And you're going to want to go ahead and first start writing out the different genotypes of each of the parents, and even they give you even child's genotypes. You can start writing those out. So first things first, they tell you a woman is type A blood, but they don't tell you if she's actually homozygous right here, or if she's heterozygous, which is this one right here, because she has an A and then an O right here genotype. So they don't tell you, they just tell you she's blood type A. So that could be either of these. So that's why I put a little dash down there. Then they tell us that they have a child with type O blood, okay, but the father is type B and he's homozygous. So they do actually tell us what he is. So the father right here is blood type B and he's homozygous. So I know he's going to be right here, this big B. That's going to be the father's genotype. So let's go ahead, finish figuring that out. Because one thing you have to recall was that the actual child is type O. So let's go ahead and figure out how we would know or not if the father is possibly that guy that she says it is. Okay, so here's the mother right here. And let's go ahead and pretend we're going to do both scenarios as if the father's homozygous and if he's actually heterozygous. So go through the whole scenario. Here's the father right here. Here's the mother as if she's homozygous for type A. So right here we have a AB blood type, AB blood type, AB blood type, AB blood type. So guess what? That child who is supposedly a type O, this won't work. This scenario right here will not work for them. So let's go ahead and try the other scenario. Let's go ahead and put the mom right here as an O. So right here, we start working this through. This right here would be the mom, this right here would be the dad, okay? So here's a B blood type child. They have a chance of having a type A child right here. They have another chance of having a type A child right here. And they have a chance right here of having a child that's type O. Okay, so you have to have both recessive alleles, which is that OO, to be considered type O blood type. So there we go right there. We know that it is possible for them and that father to be the father, but only if he was a heterozygote, not if he is homozygous. So the next kind of inheritance we're going to be looking at is sex-linked inheritance. What this one is talking about, and let me go ahead and recall and refresh your memory. Remember, we have autosomes in our pair. We have 22 autosome pairs in our chromosome makeup, and we also have one pair of sex chromosomes. If you're a female, your sex chromosomes are going to be XX. If you're male, your sex chromosomes are going to be X and Y. All in all, you should have a total of 46 chromosomes because it's 23 pairs, that's 46. Sex chromosomes are going to be our XX or your XY if you're a male. If it is considered sex-linked, that's just saying that it's actually carried on one of those sex chromosomes, either X or Y itself. A male is considered to be hemizygous because they only have one X, hemi meaning one. Now, we just finished talking about sex-linked chromosomes, but let's get a little bit more specific, okay? Let's say that the gene that you're interested in looking at is actually carried on the X chromosome and not on the Y chromosome. If that's the case, you would actually say that it is considered to be X-linked just because that's where it's carried. And a fun fact to know is that the females are actually responsible for carrying a ton of more genes on their X chromosomes versus on the Y chromosomes that make up the male. It's just literally because of size. The Y chromosome is actually about 100 times smaller than the X, so we can literally carry a lot more on the X. That's all that is. With that comes different conditions that can be passed on from the mother to the actual son itself. These would be considered recessive sex-linked conditions. One of those is male pattern baldness. We have hemophilia, color blindness, and the muscular dystrophy. Females can actually carry the affected allele, but because we have two X's, we may be considered a carrier and not phenotypically show up with that disorder or abnormality. So right here, let's say this is like complete dominance, what we originally talked about as our first mode of inheritance. So let's say a female has this normal X and then this abnormal X with some sort of disease, let's say hemophilia. So what's going to happen, most females are going to be carriers where, let's say I'm carrying this recessive allele, but since I have this one that dominates that one, I'm going to look phenotypically normal and I'm not going to have any of the actual symptoms that would come along with that disorder. And right here you can look at it and it's actually broken down very, very simply for you all to look at for a little bit more clarification if need be. So let's look at an example of a sex-linked problem. A female with no history of hemophilia marries a man with hemophilia. How many of their children can be expected to be hemophiliacs? Let's go ahead and start by writing out the genotype of, let's say, the female. So we have an X because she's a female, and then right here, remember they said they have no history of hemophilia. So she's definitely not a carrier. She's definitely going to be homozygous dominant. She does not have hemophilia at all. She is going to be crossed with this male right here. Here's one X that he gets, and he actually does have hemophilia. So right here, we're going to give him a little H for hemophilia. And here's his Y. The Y does not carry the allele for hemophilia. That's why there's no allele right here for it. Let's go ahead and walk through the cross, doing the Punnett square. So here's one gamete for my female, the other gamete for my female. Here's one gamete for my male, and my last gamete for my male is going to be my Y chromosome. So let's go ahead and process the entire Punnett square together and look at the results of our offspring. So one thing you want to notice is right here we have two X's, so I have 50% chance of having a girl and 50% chance of having a boy, just like in real life. So let's look at this. This female right here, she has a big H and a little h. That means she's a carrier. She doesn't have hemophilia, but she is carrying the allele. Here's another one. She's also a carrier. So we have two female carriers, and we also have a normal male and another normal male. So the chances of their children expecting to be hemophiliacs is going to be 0%. The only thing they have is two female carriers in that case. So let's look at another scenario. And you guys are going to go ahead and work on this one. Go ahead and pause it and try to work this out on your own. A female whose father had hemophilia marries a male with no history of the disease. How many of their children can be expected to be hemophiliacs? So, I know it sounds very similar to the last problem, but it's not. Look at the very beginning of the question. A female whose father had hemophilia. So here's our female right here. And guess what? The only thing that her dad could have passed on to her is that one X chromosome. Because this one would have come from mom, let's say, and this one would have come from dad. Because dad is X, little h, and a Y. And so the only thing he can give a daughter is that X. So he actually passed on this allele to this woman right here. So a female whose father had hemophilia marries a man with no history of the disease. So let's go ahead and look at this now. So we have a carrier female crossed with a normal male. How many of their children can be expected to be hemophiliacs? So like I said, if I were you, I would pause the video, take a second, try to go through this cross on your own and see if we come up with the same outcome. Go ahead and set up your Punnett square. Right here is going to be my female and her gametes up top, and right here will be my male on the side and his gametes as well. Once you run through the whole cross, I hope we go ahead and have the exact same outcome. But this is my results. Right here I have a female, so one female is normal, one female is a carrier for hemophiliac allele. Right here are my two males. I have a normal male, and guess what? Here we go. Lo and behold, we actually have a hemophiliac male. So the question said, how many of their children can be expected to be hemophiliacs? So here's 1, 2, 3, 4 of their offspring. So 1/4, which is this one down here, is expected to be a hemophiliac. 50% of their sons will be hemophiliac, and 50% of their daughters will be considered carriers. So let's look at another vocabulary term for you all to know. Polygenic inheritance. A polygenic trait is where there's alleles of two or more genes that determine one single trait. And epistasis is when one gene interferes with an expression of another. So in the example I have right here is the baldness gene itself. So normally what happens is anybody who has blonde hair would be, two alleles. Anybody who has red hair might have another mix of two alleles. Let's say this is going to be our big A, little a, and then let's say this one is a recessive, so little a, just for an example. Even though these are only going off of two different alleles and they're, completely dominating one another, when it comes to epistasis, there's going to actually be another allele that comes in, because remember, polygeneity is an alleles of two or more genes. So instead of having just two alleles like what we've been practicing this whole time, this one has three alleles that affect the outcome that's expressed on an individual. So let's say for this case, we actually have an allele that comes in, and when it's actually seen, it actually completely blocks out whatever the other two are doing and makes them bald. So let's say this new allele is allele E. So big A, little a, and little a, doesn't matter what's there. If there's actually right here, let's say the letter E or the E allele come in, doesn't matter. It's going to make both of these individuals bald rather than their normal blonde hair or red hair. And when we're talking about non-Mendelian inheritance, we also have to remember what pleiotropic genes are. What these are is they actually influence multiple traits. So this whole time also we've been talking about one gene, let's say the gene for hair color. One gene for hair color has two alleles, and that's all it's going to influence. So if I have big A, little a, I might have brown hair. If I have big A, brown hair. If I have little a, then I might have blonde hair. But in this case, it's different. That one single gene itself will actually affect multiple different traits. So in this example, I actually use sickle cell anemia. What happens here is right here, this gene is actually a pleiotropic gene. So instead of just coding for one particular trait, it's actually going to affect the blindness of an individual. It's going to cause them liver failure, and it may even lead to a heart attack. These are multiple traits from that one gene. That's why it's called a pleiotropic gene itself. It affects multiple different traits. All right, and that's the end of this portion of the lecture. If you have any questions, please feel free to email me, ask me questions, or on the discussion board, there's actually a place where you all can just, once again, ask any kind of questions freely. You guys can answer one another's questions in case maybe you have a better way of explaining it than I did. It's just a place where we can help each other. And if you have a question, I'm pretty sure a lot of other people in the class have the same exact question, maybe just a little too shy to ask. So feel free to ask any questions you all have.